Thank you, and morning everyone. We're a UK publicly quoted company, so there's the obligatory disclaimer statement for you. In the 15 minutes that I'll have, I'll just give you a very brief overview of ReNeuron as a business, and I'll focus on the two core cell therapy programs that we have. We are a UK-based company. We have sites in South Wales in the UK and also Boston here in the US. And we have been developing allogeneic cell therapies really since the foundation of the company. So we are most definitely believers in the allogeneic approach, using cells as therapeutics. We have two clinical stage candidates, and I'll take you through each of those in turn. We also have an emerging exosome platform, and those are exosomes derived from one of our clinical cell candidates. But I won't have time to take you through that program today, but happy to answer any particular questions you might have on that platform outside the talk. So the two core platforms that we have are given here. The first one is our HRPC platform, which stands for Human Retinal Progenitor Cells. These are retinal progenitors, cryopreserved in terms of their formulation, and targeting retinal degenerative diseases. The first target is Retinitis pigmentosa, which is currently, and this candidate's currently in a phase 1/2A study here in the US. The second asset is our CTX Cell Therapy candidate. This is an immortalized neural progenitor cell line. The HRPCs are actually expanded in low oxygen culture, so are non-modified, but the CTX cells are immortalized. And again, in a similar way to the HRPCs, they are cryopreserved, which gives them a long shelf life and thereby allows for straightforward shippage and storage and use at the clinical site when required. Both of these assets are actually partnered with Fosun Pharma for the Chinese market exclusively, and that was a deal we signed earlier this year, and we're delighted to have Fosun as a partner in that territory. And as I mentioned, we have a CTX-derived exosome program emerging, and we're primarily focused on the use of those CTX-derived exosomes as a delivery vehicle for delivering advanced drugs, oligonucleotides, RNAi-based drugs. But again, I won't take you through that today in the interest of time. The pipeline you see here, so as I mentioned, the HRPCs are targeting retinitis pigmentosa, and we're about to deliver an update actually on the data, which I'll summarize for you briefly in a second, in terms of the phase 2A part of that study. Our CTX cells are targeting stroke disability, and again, I'll come back to that later. That's currently in a phase 2B study, again here in the US, in around 30 centers, and we'll report, we hope, in the first half of calendar 21. So just looking at the HRPC program in a little bit more detail. So HRPCs are retinal progenitors, retinal progenitor cells. They are multipotent cells, so have the ability to differentiate into the cell types that are lost in diseases or retinal degenerative diseases at the back of the eye. So we know these cells can differentiate into rhodopsin positive cells and also cones. And we also know preclinically that they have the ability to engraft as well. So we're hoping that we can ameliorate the effects of retinal degenerative diseases through those putative mechanisms. We also believe the HRPCs have the ability to provide trophic support to existing surviving photoreceptors as well. We spent a good deal of time working with collaborators, including the founding institution that we licensed this technology from at Scapens, Mike Young's lab, part of Harvard Medical School, and also Moorfields in London and other institutions, really to optimize, well, to generate the IND-enabling data and to optimize the CMC characteristics of this candidate. And as I mentioned, it's cryopreserved and therefore allows for relatively straightforward expansion, shippage, and use when required. The initial target is retinitis pigmentosa, as I mentioned, which, as I'm sure you know, is an inherited retinal disease. It's a relatively large orphan status indication, around 1 in 4,000 incidents here in the US. And it's essentially a collection of many individual gene defects which collectively form the disease retinitis pigmentosa. Our own program has orphan drug designation here in the US and also in Europe, and FDA fast track designation. And the notion here is really using a gene-independent approach to address the consequences of RP as a disease. So a gene-independent cell-based therapy, as opposed to, let's say, Luxturna, which obviously is an approved drug targeting one particular gene deficiency. So that's the promise that we believe a cell-based approach holds in this particular indication. The images show you how the disease progresses. It's manifest initially in a loss of peripheral vision, leading ultimately to blindness over quite a large number of years in some cases. As I mentioned, this is a clinical stage candidate. We're currently running a phase 1/2a study here in the US. We've completed the phase 1 part of this study, which was a dose escalation study across 4 dose cohorts of 3 patients each, using an increasing dose of cells starting at 250,000 and going up to 1 million cells. And during the course of this study, we also reformulated the product from fresh, short shelf life to a cryopreserved formulation that we went on to use in the phase 2a part of this study. The phase 2a part is, under the protocol, allows us to treat between 6 and 12 patients. We've currently treated 10 of those 12 patients. The primary endpoint here, of course, is still safety, but also in this part of the study, we're monitoring on a number of efficacy measures, including visual acuity, visual field, and so on. And it's been run in 2 sites at Mass Eye near in Boston and Phoenix, under Jason Commander and Praveen Dougal, and we're delighted to have those KOL supporting this program. Again, we're going to be giving a bit, or Praveen Dougal will give more of an update on the data so far at the American Academy of Ophthalmology in San Francisco at the end of next week. But we're very encouraged by what we're seeing in the phase 2a part of this study so far. As you can see in the slide here, we've treated 10 patients, and those patients are at a number of differing follow-up points, as you see towards the bottom of the slide here. But we're seeing really quite a meaningful net change in letters read on the ETDRS chart, which you see on the right there, in the patients that have responded post-implantation of the cells. And the cells are delivered by subretinal injection to the back of the eye. So far, so good with this study. Obviously, the 12 patients in the phase 1 part of the study are still being followed up, and all patients in the study will be followed up for up to 12 months. As I said, there will be a more detailed update given by Praveen at the AAO in around a week or so's time. So just moving on quickly to our CTX program, again, a very, very different product here. It's still a multipotent cell, though, so a cell that has the ability to differentiate into terminal phenotypes associated with the organ of origin. So in this case, we're dealing with a human neural progenitor cell line that has the ability to differentiate into neurons and, to a more limited degree, into oligodendrocytes and astrocytes. We believe that CTX plays a key role in promoting anatomical plasticity in the brain, and certainly in the clinical work we've done so far, these cells appear to have an excellent safety profile. Neither in these studies or with the clinical work we've done with the HRPCs do we actually immunosuppress the patients. And in the case of CTX, the cell line was actually selected over a decade ago, actually, from a number of clones that we generated at that time, partly on its immune characteristics. Obviously, when one's working in the eye and the brain, there's a degree of natural immune privilege there in any case, of course. Again, CMC aspects are very important to us as a business. Not only do we need to generate great clinical data unequivocally, but we also need to have the means to manufacture and expand these cells at a cost of goods that's relevant ultimately to a licensee or an acquirer of the company in due course. And we believe we've been able to achieve that both with our HRPCs and with our CTX candidates. As I mentioned, and again, this is common in other cell types in certain indications, including in the CNS, we believe that CTX, and what we know from our published preclinical work, is that CTX promotes plasticity in the brain. It does this, we believe, through a number of key mechanisms, including neurogenesis, angiogenesis, and so on. So this is really providing trophic support. And interestingly, and again in common with other approaches, that we're not unique in this regard, it's become known that survival of cells in the brain is not a prerequisite for long-term efficacy. These cells are providing near-term signaling to neighboring cells where there is damage in the brain and promoting repair through those mechanisms. And the cells themselves actually don't survive in the long term, or certainly there's no real evidence that they survive in the longer term. But those mechanisms, probably mediated through the exosomes secreted by these cells, those positive effects are lasting, certainly in terms of the preclinical and the early clinical work we've done. So why are we targeting stroke disability? It's a very tough indication. It's also a completely unmet medical need in terms of patients who suffer a stroke, survive it, and have a residual motor deficit. And that's the patient group we're looking to target with CTX. So if you survive a stroke, you've got a good chance of being left physically disabled by it. And your physical disability, which is usually a unilateral deficit down one side of the body, upper and lower limb, will be manifest by around 3 months and will have largely stabilized by that point. And certainly by 6 months, you'll have a stable level of motor deficit. Patients can go on and make further cognitive improvements, but it's really the motor deficits in these patients that we're looking to address with CTX. And a way of measuring motor deficits or dependence or disability in these patients is what's known as the modified Rankin scale. It's a classic measure of disability used in disability studies. And as you can see here in the chart, and actually this is taken from a registry study published a couple of years ago in Sweden in over 40,000 disabled patients, you can see that modified Rankin as a 5-point scale measures the level of dependence in these patients. The higher you are up on this scale, the more dependent you are. And what's been highlighted in the box here is really where you see the greatest pharmacoeconomic benefit. And apologies, you probably can't see the labeling on the axis here too well, but what it shows you is the reduction in annual healthcare costs of looking after disabled stroke patients if you can drop them down one measure on modified Rankin. So as you can see, the sort of biggest bang for your buck, if you like, appears to be where patients are dropped from MRS 4 to 3 or 3 to 2. And that's a very important learning for us because modified Rankin is where we got the best response in our phase 2A study in stroke patients using CTX, and modified Rankin is also a primary endpoint, and actually the FDA's favored primary endpoint in our current ongoing phase 2B study here in the US. This slide is really just a very brief snapshot of what we saw in the phase 2A study, single-arm study in 23 patients, and it actually followed an 11-patient phase 1 study, which was eventually published in The Lancet. The data here is going in for publication shortly, we hope. And what was important in this particular study are the responses we saw on modified Rankin. So across all subjects treated, a response rate of somewhere between 27 and 35% is itself interesting. But in a pre-specified group of patients that had no residual upper arm movement going into the study, which perhaps suggests a lack of surviving cortical spinal tract, if those pre-specified patients were excluded, we see a much greater response level. And that was kind of what we expected to see, actually. So if those patients, essentially measured by having an NIHSS score of less than 4 going into the study at baseline, if those are excluded, then we certainly have a response rate here of very significant interest on the MRS measure, and certainly something that justifies further investigation in a large-scale study. And most importantly, given that the first 2 studies we've done were single-arm studies, what we're looking to do now is essentially replicate these phase 2A data against a placebo control group. Now, for a neurosurgical intervention, which is how these cells are implanted into the brain under a procedure that takes around 90 minutes and involves the patient staying in overnight, maybe 2 nights before they're then back at home, controlling for that is quite a challenge. But there is precedent for it, and what we're actually doing in our phase 2B study ongoing is monitoring patients either to the active or to placebo sham surgery. So this is just a quick sort of summary of the phase 2B protocol, and it's 110 subjects, 1-to-1 randomized either to the active or to sham surgery, treated between 6 and 12 months post-stroke. So again, we're allowing the patients to stabilize in terms of their motor deficits before they get the treatment. and we're monitoring them against the primary endpoint at the 6-month point post-treatment, and we will go on and monitor them beyond that as well, of course. There are a number of secondary endpoints beyond the primary modified Rankin score, as you see here, and again, these are fairly classic stroke disable or stroke study measures used in both acute and chronic stroke clinical studies. It's being run in around 30 sites in the US, as I mentioned. It's well underway in terms of recruitment, and we're expecting to report data in the first half of 21. I'm out of time, so I'll leave it there. Thank you very much.